All right, so here we are, day 3, and we're going to talk about cell transport, the cytoplasm, nucleus, and some extracellular stuff. So if you want to flip to that page where it has the day 3 notes, and we're going to finish up talking about the cell today. And we talk about the cell in this class because in order to understand some of the organ systems, we've got to know what cells are doing, and that'll make a little bit more sense later. And I am going to cover the kidneys today. So there's actually a lot to cover in this lecture. So I'm just going to go ahead and get started with it. Hopefully you have your paper out and you can fill in the blanks. So just a few vocab words. Interstitial fluid is the fluid outside of cells. So we usually talk about the fluid that's on the inside. So this is what's going to be on the outside of cells is the interstitial fluid. And it's important because it has a lot of nutritious stuff, like amino acids, which you learned yesterday, make up proteins, sugars, fatty acids, vitamins, hormones, salts, and then cellular wastes are usually in the interstitial fluid and will get taken up and excreted from the body. All right. The membrane, the cell membrane, is what's called selectively permeable. So selective meaning some, and then permeable meaning get through. So only some things can get through. Only some things can get out, only some things can get in. And usually what the cell wants is for nutrients to come in and waste to go out. That's just kind of a general rule, but we have other things that happen that involve the going in and coming out of things that aren't nutrients or waste. And I'm going to go through some of that transport in just a minute here. All right, so things can go through the cell membrane either passively or actively. So if it's passive transport, it means it doesn't require any energy. It just naturally happens. And if it's active transport, it's going to require energy. It's not something that would naturally happen. And so energy is required to make it happen. I'm going to give you some examples in just a little while. We'll just keep going through these vocab words. So passive transport, let's go a little bit more into detail. So remember I said passive transport doesn't require any energy. And the reason why it doesn't require any energy is because molecules are moving down a concentration gradient. They're moving from a high to a low concentration. And so the example that I usually use to help students is if I get a bottle of perfume and I spray it on myself, eventually 5 meters away from me, a student, if they're even that far away from me, let's say they're doing a lab, they're in the back of the lab room, eventually they'll smell the perfume. Well, the reason why that happened is because the perfume was very concentrated on me, and in order to establish equilibrium, the molecules spread throughout the room. They went from a high to a low concentration. And so that's our definition of diffusion. It's just the movement of molecules from a high to a low concentration in order to establish equilibrium. And that's kind of what you see going on in this beaker right here. It looks like this is some kind of tablet that's getting dissolved and the color spreading throughout the water. Right, we have simple diffusion. Simple diffusion means something is just diffusing across the cell membrane. Doesn't need any help, just goes across the cell membrane. So you have a lot of it on the outside, a little bit of it on the inside. So the natural tendency is for some of it from the outside to move into the inside in order to establish equilibrium or balance. What is going to do that? So what is going to easily diffuse across the cell membrane without any help are going to be your lipid substances. So remember yesterday when we talked about lipids, we talked about the fatty acids, cholesterol, sex hormones like testosterone and estrogen. Anything that's a lipid is going to easily move across the cell membrane. And the reason why is because the cell membrane is made up of this lipid bilayer. So if something is lipid and hydrophobic, it's going to pass through something that's lipid and hydrophobic. It's not going to be repelled by any charges or anything like that. So we're still talking about things that you would get in a biology class. So once again, those of you, I know I keep saying this, but those of you that haven't taken cell bio, this may be something that's confusing, but just get what you can from it. And we'll just kind of look at it. If you need any extra help, let me know. All right, Small molecules are going to also be able to pass through the lipid bilayer. So oxygen can be considered nonpolar, and you'd have to take chemistry to know. Nonpolar just means there's no charges on it. Carbon dioxide is nonpolar, there's no charges on it, but they're actually both really small. So oxygen and carbon dioxide can move across the lipid bilayer. All right, if it is not small, if it is not nonpolar, let's say it does have a charge on it, and if it is not lipid soluble, it's going to need a protein to help it move. So in order to get across the cell membrane, it is going to need a protein. If it is large, if it is polar, meaning it has a charge, right? If it is not lipid soluble, it's going to need a protein to help it. And here are examples of things that are not, that are polar, that are large, and that need some help. So glucose, sugar is going to need help, amino acids. Water, now water is small enough that it can diffuse without a protein, but it doesn't go fast enough. It's going to need a protein to help it move in large quantities if that's needed. And then ions. An ion is an atom that has like an electrical charge on it. So hydrogen with a positive charge would be an ion. Sodium with a positive charge would be an ion. So anything that's charged, it would be repelled by this lipid bilayer, is going to need a protein to help it get through. And that's called facilitated diffusion. So it's getting help. Osmosis is the diffusion of water. So it's the same thing we've been talking about, going from a high to a low concentration in order to achieve equilibrium. But this is specifically dealing with just water. And so water is going to diffuse freely without the need of energy into or out of the cell through a protein called an aquaporin. So an aquaporin is a protein embedded in the bilayer, the lipid bilayer of the cell membrane, that allows water to freely diffuse into and out of the cell. And that process is known as osmosis. So how does osmosis actually happen? So you've got this complicated pictures that are always shown. Anytime you talk about osmosis, you're going to see this picture. And this is called, actually it's called a U-tube. Not the channel, but it's a tube that's shaped like a U. And so what they'll do is they'll put a semi-permeable membrane in the middle here. And so what's naturally going to happen if there's a semi-permeable membrane that these red things can get through, in order to achieve balance, the red things will move. So you have only a few red things on the left, a lot of red things on the right. Let's pretend it's sugar. So you have a little bit of sugar on the left, a lot of sugar on the right. In order to achieve balance, that sugar is going to move this way. And then you end up with that. Cool, amazing. What happens though if you put a membrane that now the sugar can't fit through? What is this thing going to do in order to, what is water and what is the solute going to do in order to achieve equilibrium? Well, if the red stuff can't move to the left, water is going to go ahead and move to the right. So this is what you would find. And so in terms of concentration, in terms of number of red dots per area, it's actually going to end up equal. And so water moving across this membrane in order to achieve a balance is called osmosis. And this is what your cells are going to do. And so let's talk about tonicity, the ability of a solution to change shape or tone by changing water volume. So right here is a change in tonicity. So it's a change in water volume on either side in order to achieve a balance. Okay, we have three types of tonicity: isotonic, hypertonic, and hypotonic. All right, and so I want to talk a little bit about each one of those. So if you look back, I'm going to go back to this picture. If you look back at this picture, where is water moving? Towards solute or away from solute? So you have to think about, okay, which side actually has the most solute? The right side has the most solute. Water moves towards solute. So what I always tell students to remember is salt sucks. So salt sucks. Salt is going to suck water towards it. Sugar is going to suck water towards it. And so that's the reason why if you go to a restaurant and you eat a bunch of really salty food, you're then going to end up with salt in your interstitial fluid, the fluid on the outside of your cells. So where's water going to go? It's going to go towards the salt. So it's going to leave your cells and go towards the salt. So then homeostasis has gotten messed up, gotten out of balance. A signal will be sent to your brain to drink some water so that you can fill your cells back up. Okay, hopefully that made sense. I didn't have a picture to go with that, but I have some pictures. They're coming. All right, I just want to look at this picture right here. So what this is showing you is water is in blue, solute. Let's pretend this is salt. Water moves towards the solute. So whichever side, inside or outside, has the most solute, that's where water is going to move. And this can pose some problems. So let's look at some situations here. All right, so the place where that we're going to talk about is within the blood, and we're going to talk about tonicity of red blood cells. All right, so you have a red blood cell, and it's isotonic. So if we look back, isotonic means an equal concentration of solute on the inside as well as the outside. Well, that's nice. Water is constantly moving in and out, but do you have a net movement of water? Is water rushing out towards solute, or is water rushing in towards solute? No, it's equal on both sides. So you have a happy red blood cell. This is the reason why when you go to a hospital and they give you an IV, they are not going to infuse your blood vessels with plain distilled water, because you have things on the inside of your red blood cell. You have solute on the inside of your red blood cell. It's a cell, so it's going to have things like amino acids and some carbohydrates and all of that stuff on the inside. So if you put pure water on the outside of this red blood cell, water's going to rush in and it's going to explode. So you got to make sure as a physician or a nurse that you're going to infuse somebody with an IV bag that has some solute in it, so that you don't create an environment where water is going to make a drastic move. And so that's why when you look at your IV bag, if you've ever had one, you look up and you're like, what are they giving me? It will say 0.9% saline or NaCl. Sometimes they'll use a dextrose solution, like a sugar solution. Same thing, it's both solutes. It's both something that water is attracted to. And that's the reason why, because your red blood cells would explode if they did otherwise. All right, so hypertonic means that the cell is going to lose water. So hypertonic is what's happening when you eat a big salty meal and you have a bunch of salt on the outside of your blood cells. Water rushes towards that salt and it leaves the cell shriveled. That's hypertonic. And then the opposite would be if you have just pure water or less solute on the outside, more solute on the inside, water's going to rush in. That's a hypotonic solution. So the rule of thumb is wherever there's more solute, that's where water's going to go. And so if you can kind of visualize here it's equal, whatever, no big deal, that cell's happy. Here, there's more solute on the outside, water rushes out, the cell shrivels. Here, there's more water on the inside, water rushes in, and the cell can burst. Okay, so here we have some isotonic red blood cells and hypertonic red blood cells. So you'd have to be like severely dehydrated in this situation for your red blood cells to become like that. All right, hopefully that made sense. Let me know if it didn't. I've got to move along here. Let's talk about active transport. Active transport is where energy is needed. So we said earlier that passive transport is going to occur when something goes from a high to a low concentration. It moves down its concentration gradient. So let's use the perfume example again. So I spray the perfume on me. It diffuses across the room in order to achieve equilibrium. The student across the room smells it. What would be the opposite of that? If there was some way the student hated my perfume so bad that they figured out some way to push the perfume back to me so that they didn't have to smell it anymore. That'd take a lot of energy, and I have no idea how they'd do that. They'd have to have some kind of crazy perfume catcher. But anyways, I think that's a good example because that's a really hard thing to do. That would be kind of like active transport. You're going to push something in the opposite direction that it wouldn't naturally go in. And that's going to require ATP. ATP is energy. So you're moving a molecule against its concentration gradient from a low to a high. You're making it go backwards of what the universe would naturally want it to do. And so when something is an ion and it gets pumped against its concentration gradient, we call that a pump. And it's usually going to be a protein embedded in the cell membrane that pumps it against what it naturally wants to do. All right? So in this example, it looks like potassium's getting pumped in where it naturally would want to go out, but you're pumping it in, as much potassium in as possible. Sodium's getting pumped out against what it naturally would do, as much sodium on the outside as possible, and then it's showing that it's going to take ATP to do that. So that is what active transport is. The sodium-potassium pump is the example for active transport that you actually do need to know for this class, because a neuron could not fire unless the sodium-potassium pump set it up to fire. And so I'm going to run through this, and then I put a little link underneath this picture where you could actually watch an animation of this. And I think it's important to watch an animation because, yeah, I just think it's really important because this is kind of a difficult concept. So let's just start. I'm going to run through it. You can watch the animation if you want to, if you don't understand when I run through it. And then I'm going to run through this again whenever we get to neurons, because at that point, you do kind of need to understand what's happening here. So you have a protein and it's embedded in the cell membrane. And this is showing you a series of steps. 1, 2, 3, sorry, 4. I naturally wanted to go down here. Let me start over. Hold on. 1, 2, 3, 4, 5, 6. So it's showing you a series of steps. In the first step, this protein is taking 3 sodiums, all right? 3 sodiums. And it's pushing them, and it's using energy. So 3 sodiums bind, all right? 3 sodiums are bound. ATP is getting used for that. And then what do you see happening? It's opening up and releasing the sodium to the outside. So what's happening right here is energy is being used to pump 3 sodiums out, where they naturally, if you look at the comparison of yellows here and yellows here, the natural tendency is for these ones to come back in so they can create balance. But this protein's saying, nope, I'm going to use ATP and I am going to pump out as many sodiums as possible. So 3 each time. So this protein is now open. Now what does it do? It takes a potassium, then it takes another potassium, and then it's going to expel those 2 back in. So naturally, if you looked at this picture, where do the green dots want to go in order to achieve equilibrium? They want to go out. But the sodium-potassium pump says, no, this is active transport. Potassium, you're coming in. And so what I say, and I usually do this little hand gesture, 3 sodiums out for every 2 potassiums in. So then where do you have most of your positive charges? If 3 sodiums go out for every 2 potassiums that come in, remember you have other ions floating around. You have chloride ions floating around, and chloride is negatively charged. So if you mix all of that in here, what are the net charges on the outside and the inside? If you have more positive on the outside than you do on the inside, then you have a difference in charge across the cell membrane. So what the sodium-potassium pump does is it causes the outside of the cell to be positive and the inside of the cell to be negative. And that sets that cell up to do a certain kind of work. And I hate to leave you hanging on that, but that is how a neuron gets set up to fire. So if you didn't understand that, go to the tiny URL and watch the animation if you want to. If you want to just wing it and you want to wait until I talk about neurons to do this again, I'm totally fine with you doing that. But I just want you to know the sodium-potassium pump requires energy. It pumps sodium out, potassium in against the concentration gradient, and it achieves having a positive charge on the outside and a negative charge on the inside of the cell, setting it up to do a certain kind of work. Right? The other type of active transport is going to be vesicular transport, and that's going to use vesicles, which uses the membrane. So an example of this would be a white blood cell engulfing a pathogen. It's the pathogen is too big, or the substance is just too big, so the cell membrane itself forms a little bubble or envelope around the substance, bringing it in. That's endocytosis, bringing it in. And then exocytosis is when you have cell membrane that is going to fuse and it expels the content. All right, so this is just with vesicles. This is using the whole membrane. This isn't using transport proteins, it's using the whole membrane. Endocytosis and exocytosis. All right, the cytoplasm, just a little bit on the cytoplasm. And yeah, we still have a lot to cover here. So the cytoplasm is between the plasma membrane and the nucleus, and there's three elements. You have the cytosol, which is the fluid, and that's going to have water, protein, salt, and sugars. All right. You have organelles. Remember, organelles are going to have specific functions. And I'm not going over all of those because that's cell biology stuff. And then your inclusions are going to be chemical substances that vary on cell, depending on cell type. So some cells are going to have more mitochondria than others. Some are going to have more endoplasmic reticulums than others. I wouldn't focus a lot on this. I just really included this page for completion's sake. I'm probably not going to ask you anything on it. I just put it on there because cytoplasm is part of the cell. So I needed to just say something about it. So I'm going to kind of skip past this. Hopefully I'm not going too fast for you. I want to go ahead and get to the kidney. Okay, just for completion's sake, I'm going to tell you a little bit about the nucleus. The nucleus is the control center of the cell that contains DNA. Most cells have only one nucleus, except muscle and liver can have multiple nuclei. And then some cells have no nucleus at all. Mature red blood cells, there's just no point. There's no reason to, because what they're really doing is just carrying oxygen around. So there's really no reason to. So the three main structures of the nucleus are going to be the nuclear envelope, which covers the outside, the nucleoli, which is that's the plural form of nucleolus. So just so you know, nucleoli, nucleolus, I'm talking about the same thing. That's the very center of the nucleus, and its job is to produce ribosomes. And ribosomes are, remember, going to leave the nucleus and they're going to be attached here. And ribosomes are where proteins get made. And then you have chromatin. Chromatin is your DNA, packaged DNA. Right, and then it's just showing you multinucleated muscle cells, multinucleated liver cells, and then anucleated red blood cells. So the reason that muscle cells and liver cells are going to have multiple nuclei is because they produce that much protein. So protein production is so much more ramped up, and it happens a lot more at a faster rate in muscle and liver. And remember, muscles for movement, and muscles are made of protein. Liver is going to have to do with detoxing, and it uses proteins to do that. Your red blood cells are just carrying oxygen, so that's why. So just in case you're wondering, that's why some cells have more nuclei than others. Okay. We're just kind of breezing past this. I hope I'm not going too fast. I'm just going over the parts of the cell. And it's all written there for you. I don't really have a lot of fill in the blank. And you're just going to reference this when you take your little quiz. I think y'all saw yesterday that it's all in the notes. It's all in the notes. Chromatin is, like I said before, that's the DNA. And it's wrapped around these little proteins called histones. DNA gets wrapped around proteins. And all it is just a packaging thing. DNA gets packaged, it wraps around these proteins so it's more easily packaged and it's organized. It's like you wouldn't just throw all your clothes in a big barrel. You put your clothes in drawers, right? It helps organize it. Hopefully that was a good analogy. I have no idea. But it helps organize it because there's so much DNA. All right, so chromatin is DNA with the proteins it's wrapped around, your little dresser drawers right here. And then the nucleosome, this one little unit right here, is called a nucleosome. This one little unit. All right. So you have a protein with DNA wrapped around it. So the nucleosome, like in my analogy, would be the drawer of the dresser. Right? And then histones is the name of the protein, the little blue things that it wraps around, and it just allows for compact and orderly packing of DNA. So during cell division, chromatin condenses and forms chromosomes. So this is where chromosome comes from. Chromosome is DNA, but it's DNA tightly packaged with that histone protein. Right? And that just makes sure that things don't get all loose and tangled up and not a big mess. So it's very organized. And so what happens with DNA inside of the nucleus is that it's going to be copied. And it's a process called replication. And so I know I'm touching on a whole bunch of different topics here, but really we're looking at the cell, we're looking at the contents of the cell, and instead of going through each organelle, I'm just talking about the nucleus. And inside the nucleus is DNA. DNA is packaged into chromosomes. Chromosomes get copied before a cell divides, and that process is called replication. When the cells divide, that's called mitosis. All right, so DNA replicates to form two daughter cells, and then you have mitosis, and you don't need to know the different phases of mitosis. This is just for completion's sake. DNA is going to condense. It attaches to these spindles, lines up in the middle, it gets pulled apart, and then the cells divide. So overall, all I'm telling you here is DNA gets copied, a cell divides, and each new cell has identical copies of the DNA. All right, and I'm getting to a point here, and there's a reason why I am actually talking about this. DNA is the blueprint for protein synthesis. So DNA is there to make proteins. A gene is the segment of DNA that codes for a particular protein. And so what I'm showing you in this picture is DNA is going to get transcribed into RNA. RNA is going to leave the nucleus and is going to get translated into a certain protein. All right, so what is DNA for? DNA is for the production of protein. It codes for proteins. All right. So transcription is the process by which RNA gets formed from DNA. Translation is when RNA basically gets translated into a protein. And this would take like an entire week. This right here, this picture right here, I would spend an entire week going through this. So you can imagine how uncomfortable I'm feeling right now putting it into like the 5 minutes I'm putting it into. So like I said before, take what you can, absorb what you can, and if you have trouble, we'll fix it, okay? We'll fix it. Don't stress. All right. So the reason I talked about this is, I want to make this really clear. Every single cell in your body has the exact same DNA. Every single cell. So why is it that your eyeball does one thing and your stomach does another? If those cells have the exact same DNA, identical DNA, shouldn't they be doing the exact same thing? And the answer is no. They have identical DNA, but some DNA is expressed in your eye and other DNA is expressed in your stomach. So just because your stomach cell has all the DNA, it's not using all of it. It's just using the DNA that helps the stomach cell do its job, which is to digest food and to absorb nutrients. Okay, so that's the point I was trying to make by talking about DNA is that a cell becomes specialized to do what it does, whether it's an eyeball cell, a liver cell, a stomach cell, because of the segments of DNA that get turned on. And because the segments of DNA that get turned on, proteins get made. And that's called cell differentiation. And I'm bringing that up, and I'm hoping that everyone is grasping what I'm saying, because tomorrow I'm going to talk about stem cells and the understanding of all cells have identical DNA. Some DNA gets turned off, some DNA gets turned on. then certain proteins get made, and that's how that cell becomes specialized in what it does. That's very important when we talk about the topic of stem cells. Okay, I'm going to bring this up again in the Zoom call tomorrow. All right, and we already talked about that, transcription translation. Okay, we're kind of getting to the end here. Extracellular material, any substance that's outside the cell, like body fluids, blood plasma, and when we get to blood and blood vessels, I'll talk about those. Interstitial fluid, remember it's the fluid that has all the nutrients and stuff. Cellular secretions like saliva, mucus, gastric fluids. Those are just extracellular materials that sit outside of the cell. And then the other thing that is on the outside of the cell is the extracellular matrix, and that's the glue that holds cells together. So there's a jelly-like substance that's made out of proteins like collagen and carbohydrates, and there's a picture there, and it's the thing that looks kind of like a barbed wire. The barbed wire looking thing is the extracellular matrix, and it is what holds cells together. So I just went through cell biology well, not all of cell biology, but I went through a lot of cell biology in a matter of 2 days. So if some of you who haven't taken are feeling overwhelmed, that's okay. I know I keep saying that, but I just want to, I want to emotionally rescue anybody that's feeling really stressed out about this right now. Just go with it. Just roll with it. You'll be fine. All right. So I want to finally get to body systems. That's kind of what I'm doing here because I think that's why you took this class, is to get to body systems and to learn about diseases and stuff like that. So I want to talk about our focus today, which is the kidney. So remember I said osmosis is the diffusion of water from a high to a low concentration of water. The urinary system is going to rely on osmosis and aquaporins pretty heavily. And so here are your kidneys, all right? And if you took all the organs out of your mouse the other day, you would have seen these two things that actually look like kidney beans. And you're like, what? That's where the name kidney bean comes from. I'm never going to feel this same way again eating kidney beans because it looked like just like those little kidneys found in the mouse. That's why they're called. So the kidneys are sitting in your back. And this is the reason why if someone is like an avid runner, like psycho about running, like runs like crazy marathons, they will start urinating blood eventually because your kidneys are kind of sitting in your back and they're flop around if you run too much and you can really damage yourself. So just kind of a little side, just a little pearl of wisdom there. Don't run too much or you'll damage your kidneys. So this is what the kidney looks like. All right. You have your renal pelvis right here. You have these blood vessels. So the kidney is basically just filtering blood. And then what's interesting about the kidney is that the adrenal gland sits on top of it. And when we get to endocrine system, we'll talk about adrenal gland. But basically all I want you to see here is where the kidney is located and that the kidney has a ton of blood vessels and it's going to filter the blood and it's going to pull out anything that is a waste product. And it's also going to rely on the diffusion of water to come with the waste product. The functional unit of the kidney is called a nephron. And so if you look right here, we're going to zoom in right here to the nephron, then zoom in right here. And so what the nephron is a series of tubes that are going to basically rely on the osmosis of water and waste across into the tube. So where you're going from the blood vessel into these tubes right here, these peritubular capillaries. And then all of the water and waste is going to get collected from the blood vessels into the collecting duct. And that is how urine gets made. So it relies on diffusion and osmosis to do that. So your assignment today, and I think I had like a biointeractive here, but you're not doing that. You're going to do a simulation of the kidney. And what you're going to do is you're going to get your dialysis tubing and you're going to put your simulated blood into the dialysis tube, which acts as your blood vessel. You're going to put it in water. And so this time we already have the water on the outside of the blood vessel. And what you're going to see is the water and the waste diffuse from the blood vessel into the outside to make it your kidney. Then you're going to test your blood, or not your blood, you're going to test the urine for glucose to see if sugar came out. And that's something that you definitely don't want to have happen. And I'm going to have you Google. So there's only 3 questions you're going to answer after this simulation. I'm going to have you Google why is this a bad thing for glucose to move into the urine? And then you're going to do your dialysis simulation. And the directions are very, very straightforward. And I would have talked about these simulations in the Zoom call already. So I am going to go ahead and end this video. See you guys later.